Welcome back to lesson 3 of our inflammatory process. Right now, what we're going to be spending time on for this lesson is looking at the chemical mediators that are involved in the inflammatory process and how they help aid us in trying to repair the area wherever the injury has occurred. So I do want to take the time. It's just a 3-minute video. I know we saw this in our last lesson, but I think it's always good to just kind of remind ourselves of where we're starting and moving on after that. So let's watch this video.
Inflammation is a critical innate defense in the war between microbial invaders and their hosts. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells called macrophages may wander into the infected area and engulf the invading organisms. The macrophages digest the material and in this way help clean up the infection. In response to the infection, macrophages release a number of chemicals, including those called cytokines. Cytokines are small proteins that regulate the immune response. Some diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils, snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system called mast cells that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
All right, so now let's take a look at some of these chemical mediators in a little bit more depth and talk about how they are contributing to the different hallmarks of inflammation that we experience. So first, we're going to start off with one that was just introduced in that video, which is the bradykinin. It does work to help vasodilate and increase that permeability. And we'll talk about in just a moment how that occurs. It also happens to activate pain receptors. And so part of where we are experiencing pain as a hallmark of our inflammation is coming from the bradykinin. There are also complement proteins that are released with the degranulation of the mast cells, which include things like histamine and heparin. Histamine works alongside with bradykinin in order to increase that permeability by separating those endothelial cells in order for fluid to be able to escape out of the blood vessel and into the area of injury. And then remember what heparin does is it stops any clotting mechanisms from happening so that fluid can actually exit out those gaps or those clefts. We also have some vasodilation that can come from nitric oxide, which is released when endothelial cells are activated. So, the activation of endothelial cells, because of the rush of blood flow that is occurring, it actually is a frictional force that will activate those blood vessels, the endothelial cells within the blood vessels. And it will help to release nitric oxide, which will call more blood flow into the area. Prostaglandins, we also just took a look at in that video. And prostaglandins work both on pain receptors as well as on the endothelial cells to cause vasodilation. TNF alpha, remember, is one of our activators of our macrophages. And so macrophages are the cells that are responsible for consuming a lot of the debris that is created because of the injury. And as a byproduct, macrophages being active will release TNF alpha, which will then in turn reactivate more macrophages and call more macrophages into the area. Interferon is a chemical mediator that activates the mast cells and the macrophages. And then there's a couple interleukins that are involved. Interleukin 1 activates our endothelial cells, as well as interleukin 16 activates and migrates T cells to the area to help join in the fight. Now, what I do want to point out is that these are only some of the chemical mediators that are involved in inflammation. There are actually more that, if we wanted to go into a really microscopic level and start studying it more in depthly, but I wanted to start us kind of easy on some of the main players. Now, coming back to our hallmarks of injury, when we have the swelling, the redness, the warmth, the pain, and the loss of function, what I thought we could do for just one moment is to actually look through each one of these hallmarks of injury and talk about what are the chemical mediators that are responsible for it. So we talked about the chemical mediators and what they do, but now let's take a look on how each of them are actually leading to these hallmarks of injury. So I just want to remind us of our little flow chart that we have here. So we have our tissue injury that occurs, and we have a huge release of these chemical mediators, such as like the histamines, the complement proteins, the kinins like bradykinin, prostaglandins, and all the interleukins, everything that's occurring here. And they're going to work together to be able to vasodilate, increase that permeability, and start to attract all the neutrophils and monocytes and other white blood cells to the area. So we're going to walk through and talk about how do these actually lead to the heat, the redness, the pain, the swelling, and the loss of movement that we can have. So let's take a look at our sources of tumor, which remember is that increase in swelling and edema that can happen within the area. So what we'll end up doing is increasing blood flow to the area itself, because we need to call in all of those white blood cells, the neutrophils, the monocytes, to the area to be able to help in the inflammation process by consuming all of those cells that need to be consumed to help heal the area. The chemical mediators of nitric oxide, bradykinin, and histamine all contribute to this vasodilation. Prostaglandins also increase blood flow to the area through vasodilation. And the mechanism of action on how that works is looking at the cyclooxygenase 2 receptor. So, what happens when there is injured tissue, it actually activates the arachidonic acid, which will lead to stimulation of these COX2 receptors, which will produce prostaglandins. And those prostaglandins will act in a very similar fashion as the histamine and bradykinin and nitric oxide. Now, beyond just calling more blood flow to the area through the vasodilation, we also will increase the permeability of the plasma portion of this blood flow to actually leak out into the interstitium. So edema is caused by that same action with histamine, bradykinin, prostaglandins, all opening up those gaps in the intracellular clefts, and then heparin will keep those open so that fluids and proteins and white blood cells are able to leave that area. So let's take a look at this blood vessel physiology in a little bit more depth. So we're talking about having an injury in which we ultimately have activation of our mast cells, which will release that histamine and heparin. In conjunction, because of the tissue injury itself, we have a release of bradykinin and prostaglandin. And these four chemical mediators combined will start to work on the lining of the blood vessel wall. So it takes what looks like a seemingly solid wall and it starts to contract the endothelial cells together, creating gaps in between them. And these are our intercellular clefts that we have. And we start to have a fluid and proteins leak out. Now, remember, we also have monocytes and neutrophils that are walking down the walls and being called to the area through margination. And once it comes to these intercellular clefts, we'll exit in diapededis outside into the interstitium. Now, remember, if it's a neutrophil, it will just start consuming all of the debris and all of the dead tissue that's around. However, monocytes, after we diapedese and enter into the interstitium, will actually turn into our macrophages, which will now go on to consume all of that dead tissue. So now let's take a look at our sources of rubor. So this is our redness that we can see in the area. So remember, when we have vasodilation in the area, it's increasing blood flow to the area. And as it does that, it's bringing more red blood cells to the area as well. And that actually, with that congregation, you can start to see the redness that's starting to occur. So we'll see nitric oxide, histamine, bradykinin, and prostaglandins all playing a part. Now, these substances are causing the vasodilation that causes this redness. We can also see that with our warmth. So that's also bringing, these same chemicals are bringing more blood flow to the area, and it's going to cause warmth in the area as well. So blood in itself has a temperature that is higher than what the skin tissue is. It's approximately 100.4 degrees versus skin tissue is considerably less than that. Now we add into it our edema, or this permeability changes that are happening because of the histamines and bradykinin and the heparin that is helping to keep those separations in the blood vessel between the endothelial cells open. Now what happens is plasma is actually smaller particles. The fluid is smaller than what the red blood cells are. So red blood cells have a harder time leaking out into this area, but the white blood cells and the plasma can leak out into that interstitium. With it, it is bringing the warmth of the blood itself to the skin surface, and so that's where we see our increases in warmth. Now, what I also want to note on this is that when we are dealing with an increase in warmth, the kinetic energy of that, what it does is it actually increases our rate of metabolism. And so it actually helps us to speed in the healing process. So not just is it bringing all of the great and amazing white blood cells to help come and clear up all of the debris of the injured tissue, or in the case if it was bacteria that it was trying to get rid of, it's also going to speed up the healing process because of speeding up the metabolism due to this warmth. Now, pain, what are we experiencing our pain from? So what we've already noticed is that we do have that release of bradykinin, which does stimulate the pain receptors directly. Besides bradykinin, we also have a release of prostaglandins in the area. Our injured tissue will actually activate arachidonic acid, which leads to receptors that are called COX-2 receptors being activated, and that will actually lead towards the production of the prostaglandins, which then bind to our pain receptors. What's interesting is that COX-2 is stimulated in order to produce this prostaglandin. And if you think of ibuprofen, the mechanism that ibuprofen works on to help kind of regulate pain that's being felt is it actually blocks the COX-2 receptor. And that's what the mechanism of action is of ibuprofen. So when we're taking ibuprofen, it is actually the blocking of the COX-2 receptor, which will then stop the production of the prostaglandins. And that's why we can experience less pain with consumption of ibuprofen. We also have some pain literally just because of the swelling that is occurring in the area. So pressure starts to build up because the fluid is contained and it can't go anywhere else. And so it starts to build more pressure in the tissue itself, which can start to compress our free nerve endings, and that will stimulate a pain sensation as well. Now we're taking a look at our function and our loss of function that can occur because of the injury. So we have swelling in the area, which starts to increase that pressure. And when it does that, it starts to result in a little more stiffness in the area. And it's a more difficult time in trying to produce movements. Also, the pressure on the nerves themselves, on any efferent nerves that are coming through, which are our motor nerves that control movement, when we put pressure on those nerves, it can also disrupt any kind of neural communication that's happening to come down towards muscle contraction. So, if we are disrupting this communication from the brain to the muscles because of the pressure on the nerves, we're also going to experience some difficulty in movement because of that. Also, logistically, just because we are experiencing the pain that we saw in the last slide, it actually reduces our willingness to want to move. And so we end up actually guarding the area, which means trying to protect it. And we can protect it by not moving it as much. So this concludes our lesson 3 of the inflammatory process. Make sure that you've been answering the questions down below. And then we will come back for lesson 4, in which we will start talking about how is everything cleaned up in the system after we have activated the inflammatory process. As well as prostaglandins.